हाँ हो गया।
आपकी आवाज़ कम आ रही है, उसको वॉल्यूम हाई कर सकते हैं?
प्रकाश जी यूट्यूब का वॉल्यूम हाई करो प्लीज। प्रकाश सर यूट्यूब का वॉल्यूम प्लीज रेज।
यूट्यूब का वॉल्यूम
हाँ जी उसको तेज करो।
बस ये इतना ही हो रहा है,
जी
इससे ज्यादा नहीं हो रहा है।
ओके।
Today she is sick actually. Let me find a.
हाँ सर।
आपकी आवाज़ तो आई नहीं।
ये दूसरा वीडियो शुरू हो गया।
आवाज़ नहीं आ रही, बिल्कुल भी नहीं आ रही, कोई
all of the
मतलब नहीं।
moisture out of your brain and broke it down to its constituent nutritional content, what would it look like? Most of the weight of a dehydrated brain would come from fats, also known as lipids. In the remaining brain matter, you would find proteins and amino acids, traces of micronutrients, and glucose. The brain is, of course, more than just the sum of its nutritional parts, but each component does have a distinct impact on functioning, development.
ओके, बंद कीजिए, मैं लेक्चर लेता हूँ।
So that lunch habit or late night alertness.
यूट्यूब हटा दो।
बंद, हटा दीजिए। See, gut microbiota brain axis. If you see this, the learning objective of this lecture is to understand the neurophysiological connection that enable the biodirectional communication between the gut and the brain. So there is a biodirectional connection. There is a connection between the gut and the brain, which is through the vagus nerve. Is a nerve which connects directly from the gut to the brain. And the main learning objective is to identify the lifestyle, dietary, and microbial influence on the flow of function and signaling molecules along the gut microdata brain axis. Now you can see the what is the main objective of learning this class. Main objective is to identify the dietary and microbial influence. So what are the influence of the microbiota on the flow and function of signaling molecules along the gut brain axis. So how microbiota damages the brain, you will understand through this. And independent dietary regimes that target the gut and the intestinal microbiota to improve and maintain the optimal physical and mental health. So you will also get to know about the dietary regimes by which you will help to understand the to improve the gut and brain axis. तो इस मुख्य धारा में हम समझ पाएंगे कि गट और ब्रेन एक्सिस को किस तरह से कनेक्शन दिया जाता है, किस तरह से शरीर को माइक्रोबायोडाटा कलेक्शन एंड माइक्रोब्स कैसे डैमेज करते हैं, और उसके बाद न्यूरो इन्फ्लामेशन, न्यूरो डिजेनरेटिव डिजीज कैसे जनरेट होती है, और बाकी बीमारियां भी कैसे जनरेट होती है, जिसमें दिमाग का बहुत बड़ा रोल है। इसमें सबसे बड़ी बात होती है गट ब्रेन एक्सिस को समझना होता है. So now we can understand what is the importance of gut brain axis. So now microbiome, write down the definition of microbiome. A collection of microbial genomes. एक हो गया genomes, genomes. A collection of microbial genomes. Microbiome and microbiota is a collection of microbes. Is a collection of microbes. Now you must know there are many bacteria in the body because we have life. The negative bacteria they may create disease, but after life we don't put the body in our house because these bacteria become active and they create a problem for us. As many as 150 X bacteria, means more than 150 million bacteria, bacterial genes than our human genome. We have more than 150 million bacterial genes than our human genomes. तो हमारे शरीर में ढेरों लाखों बैक्टीरिया जनोम है जो नेगेटिव भी है पॉजिटिव भी है बैक्टीरियाज आर गुड बैक्टीरियाज आर बैड नाउ इट इज अप टू आवर डाइटरी इनफैक्ट विच काइंड ऑफ बैक्टीरिया be allowed to exist. So stomach and duodenum you will have more than Streptococcus and Heptobacillus bacteria. Genuine aluminum you will have bacteria, Streptococcus, Lactobacillus, and so on. So colon also will have number of bacteria, which helps both in creation of disease and also in digestion of the food. So whenever you eat, this gut inflammation is there. This destroys the gut. Now when this gut is destroyed, what you see, the inflammation is created in the brain. So this part can be understood by the how you protect your health, and how this happens is by the microbiota functions. Write down the keynotes of microbiota functions. It helps you in protective functions. Microbiota functions include protective functions. Write down structural functions, metabolic functions, fermenting dietary fiber into short chain fatty acid, synthesizing vitamins. Protective functions, structural functions, metabolic functions, fermenting dietary fiber into short chain fatty acids, and synthesizing vitamins. So if you take a lot of antibiotics, what are the things we are going to lose? We are going to lose all these things, especially the synthesizing of the vitamins. Yani microbiota functions क्या है? प्रोटेक्टिव फंक्शन ये प्रोटेक्ट करता है, स्ट्रक्चरल फंक्शन ये स्ट्रक्चर को मेंटेन करने में मदद करता है, दूसरा ये डाइटरी को शॉर्ट फैटी एसिड्स और विटामिन्स बनाने में मदद करता है। अगर ये ठीक से काम नहीं करे तो शरीर में विटामिन से सबकी कमी हो जाती है। सो यू कैन अंडरस्टैंड द रोल ऑफ माइक्रोबायोटा। What are they? Protective functions, structural functions, fermenting dietary fiber into short chain fatty acids, and synthesizing vitamins. And this is only possible when the gut is intact. So it is possible when the gut is
Healthy?
healthy and intact. Intact means there is no leak in the gut. The gut is like this. The moment it becomes like this, it becomes a unhealthy gut. Clear?
Yes, sir.
So this gut microbiota have a connection through the vagus nerve into the various areas of the brain. It also leads to what? Protects us from various diseases. In order we can understand microbiota and G lymphatic system, write down. Microbiota and G lymphatic system is responsible for what? Helping you to protect from many diseases. So how does gut and microbiota communication take place? Write down. Bi-directional communication. It is called bi-directional communication. What it is called? Bi-directional. Anyone in the class or not? Can you hear me?
Yes,
Yes,
sir.
yes, sir.
Yes, sir.
Bi-directional communication.
So there is, it happens to the central nervous system. In central nervous system consists of two parts, brain and the spine. CNS has two parts. What are they? Brain and the
Fine.
Spinal cord.
spine. Autonomic system is autonomic nervous system is consist of two parts. What are they? Sympathetic and parasympathetic. CNS consists of brain and spine. Autonomic nervous system consists of sympathetic and parasympathetic. Now, if there is an imbalance of parasympathetic and sympathetic, can you tell me which is the major disease you see in the patient? पैरासिम्पेथेटिक और सिंपैथेटिक इम्बैलेंस हो जाने से आपको एक मेजर डिजीज दिखाई देता है, बता सकते हो उसका नाम क्या है? Anand, can you put a light on that the disease?
Autoimmune.
नहीं, intentional tremor. Autoimmune is created by the gut brain toxicity, and this is created by the what? Imbalance between the
Parasympathetic and sympathetic
autonomic
nervous
nervous
system.
system, which consists of parasympathetic and sympathetic. And it always happens when there is more stress in the patient. Stressful life creates intentional tremor. These tremors will increase when there is a what? Much stress inside. So there is another system called enteric nervous system. Is the intrinsic nervous system of GI tract. This is called the gut tract. The third system is what? The enteric nervous system, ENS, enteric nervous system. What is the third system? It is enteric nervous system. Then comes the hypothalamic pituitary adrenal axis, HPA, hypothalamic pituitary adrenal axis. And fourth is microbiome, collection of microorganisms and their genomes in the gut. What are the first thing? Gut microbiota brain communication. So we should understand what is brain communication first. It is first the CNS communication, which is brain and the spine. Second, autonomic, sympathetic, parasympathetic. Can you open only one mic? Nigeria, Paul, mute your mic. Only one mic to communicate with me. Anyone can open the mic. Whose background is not noisy. Anyone? आनंद आप अपना माइक खोल दे.
Sir, background is very noisy, sir. But try it.
अभी तो नहीं आ रहा इतना आवाज.
Okay, sir.
चलो. So bi-directional communication, दो directional communication होता है. तो first is the what are the communications level. तो जो सबसे पहला communication level होता है वो central nervous system का होता है. Central nervous system consists of the brain and the spinal cord. Am I clear? Second consists of the autonomic nervous system. Autonomic nervous system consists of parasympathetic, sympathetic. Then of the enteric nervous system, it is through the gut. Hypothalamus pituitary adrenal axis. Hypothalamus pituitary adrenal axis. What will you write? Hypothalamus pituitary adrenal axis. And microbiome, collection of microorganisms and their genomes in the gut.
Sastri ji, I'm fully back now with host. You still want the video?
Yeah, you can put up if you are clear with that. You are still on the same part.
the gut and the brain communicate has fascinated us for centuries. The gut-brain axis transforms information via the vagus nerve from food to feelings. Once eaten, digested food particles enter the small intestine, which is covered with a velvety layer of villi. Each villus is lined with a single layer of epithelium. This layer is made up of different cell types. One of them, the enteroendocrine cell, is unlike the others. It is our gut sensor. In addition to communicating through hormones, we discovered that enteroendocrine cells also synapse with nerves, including the vagus nerve. We call those enteroendocrine cells synapsing with nerves neuropod cells. They sense and react to their environment. They sense mechanical, thermal, and chemical stimuli, such as nutrients or bacterial byproducts in the gut lumen. Inside neuropod cells, signals from stimuli are converted into tiny electrical pulses. These pulses propagate via synapses onto the afferent neuron of the vagus nerve. Vagal neurons carry the sensory information to the brainstem, linking the signals generated inside the small intestine to the brain. The neuropod cell connection with the vagus nerve serves as a conduit for food in the gut to influence brain function within seconds. This connection is also a potential portal for gut pathogens to access the brain. This new knowledge is a foundation for designing therapies to treat disorders related to altered gut-brain signaling.
So, did you understand how important this communication level is? The most important part is the vagus nerve. We already told you. So, it influences various symptoms and various diseases through the genomes, negative bacteria generated in the gut. Now, that is why we should keep our good eyes on our food, whatever we eat. हमें अपने खाने पर बहुत ध्यान देना चाहिए. That is why I mentioned the paleo diet yesterday. That you should be careful about what? 4 things in most of the diseases. What are they? Sugar, artificial sugar, gluten, dairy, and
Preservative item.
preservative items are always there. And
Artificial sugar.
pardon me?
Gluten, sir.
Artificial sugar.
So non-veg. Non-veg.
You are right.
Artificial sweeteners.
Sea salt, sir.
So, many replies are there, and these are all correct replies. Sea salt, sodium chloride is one of the very commonly known cause for what? Essential hypertension and other diseases, which again influences the vagus nerve, and also vagus nerve has influence on the heart. So, the more you take the sea salt, the more chances you have of having hypertension. So, instead of taking sea salt, which is having higher quantity of sodium chloride, you should take potassium chloride. Gluten has to be avoided.
Do you want the second video, Sastriji?
Yes. We'll go to the second video. Before that, we like to put up the neurotransmitters which helps in generating our acetylcholine, noradrenaline, adrenaline, GABA, serotonin, and dopamine. They are generally influenced from the gut-brain axis. What are the neurotransmitters? Acetylcholine. Now you will see many patients who are suffering from gut inflammation, most of us suffering. You'll see at a very young age the hair becomes white. Hair fall system starts in them. It is because of the gut inflammation. Depression starts. Why? Because of the neuroinflammation. Why it is? Because of the miscommunication between the gut and the brain, and it happens because of the bacteria generated in the brain. So what are the hormones illustrated? Acetylcholine, noradrenaline, adrenaline, GABA, serotonin and dopamine. So you can play the second role.
Hello, I'm Ruslan Majitov. Today I will talk about the role of gut-brain axis in food allergy. All diseases are abnormal versions of some normal physiological process of function, and to really understand a disease, we need to know what the normal physiological counterpart is. And in particular, many diseases are due to excessive or insufficient expression of various body defenses. In the case of allergies, I think the normal counterpart of allergic disease is what I refer to as allergic defense. Allergy is typically thought of as a mistargeted pathological response to innocuous environmental substances, such as food proteins. However, all allergic reactions exhibit the features of defense systems that protects us from noxious substances. All symptoms of allergy, such as running nose, sneezing, itching, vomiting, diarrhea, have to do with expelling something from the body. So therefore, this defense from noxious substances, I think, is the normal counterpart of allergic disease. So in the case of food allergy, the normal counterpart would be, again, something that protects us from potentially harmful substances in the food. So food allergy is a reaction of the immune system to food substances that our body, for one reason or another, deems to be noxious, harmful to us. And normally, food allergy is a part of what I call a food quality control system that includes olfaction, taste, and various gastrointestinal chemosensing mechanisms. And pathological food allergy then would be an overreaction of this quality control system. The quality control for food is important because food is not just nutrients. All food sources, natural food sources anyway, contain a variety of chemicals. Some of them are innocuous, some of them are useful for our bodies, and some of them are harmful. And how our body deals with this huge diversity of thousands of different chemicals is largely unknown. And here on this slide is just a snapshot of some of the chemicals identified in some of these common foods. And for the most part, we don't know how any of these molecules are perceived by our body, whether they actively sense, and what are the consequences are of exposure to many of these chemicals. And plants may make many chemicals for defense purposes to protect from being eaten by herbivores. And so a lot of plant secondary metabolites are actually designed to be toxic to animals. And therefore, it's very important for animals that consume plants to be able to detect these noxious chemicals and to react accordingly. And plant-derived chemicals is only one example of this rich diversity of molecules that are present in normal foods. And we actually know very little about the full universe of chemistry that we're exposed to, both natural chemistry as well as chemistry that is a product of industrial production, such as the one that's present in modern foods during food processing, in detergents, and other chemicals present in normal household items. So the way that food quality control works, starting with more familiar examples, before we even consume something, it has to pass a smell test. So if it smells fishy, if there is something we don't like about the smell, we don't ingest it, obviously. But if it smells okay, we proceed with ingestion, and at that point, the gustatory sensory system detects the quality of food based on its taste by detecting various molecules present within the food. And again, if it tastes in a way that is suspicious to us, we don't proceed with ingestion. But if it tastes fine, we swallow, and then the food gets into the GI tract, where there is
Can you
another
stop it here?
chemical sensing system that evaluates the quality of food. Most of what we know
Can you
about
stop
this
it here?
chemosensing system is based on recognition of macronutrients.
So we can see by now we have understood many things about the gut brain axis. It's a very long chapter. We'll have to have a long discussion because this is a very new field about which even the neurologists and others they do not know, and because of their wrong decisions while treatment, people suffer from large number of problems. Now, number one, number two, number three. There are three things which we must understand. To keep our gut healthy, we must maintain the pH balance of the water. Now, the water which we are drinking is less than 5, while the blood pH balance is more than 7.5. So, now the body has to go with 7.5 and excess use its own minerals to maintain the TDS quality, in which the gut is very seriously harmful. Gets more what? Chances to generate negative microbes. Yeah. So God has given us what? Many things to sense whether the food is correct or not. The first is the olfactory. So you smell the food and then you taste it with the what? Tongue. Now various molecules in the tongue taste the food and then realize whether the food is correct or not. यानी पहले आप नाक से सूंघ कर खाते हैं, सूंघते हैं उसको, उसके बाद जीभ पर रखते हैं. तो जीभ और नाक का जब संतुलन बैठता है कि हाँ सब कुछ सही है, तब आप उसको चबा के खाते हैं. वरना ब्रेन इज गुड इनफ टू अंडरस्टैंड द फूड इज what vicious and poisonous. Go ahead.
Proteins, carbohydrates, and fats. And there are a variety of specialized sensory epithelial cells that detect these macronutrients and report their value to the brain and to endocrine cells through production of various gut hormones. This communication is very important for adjusting our metabolism, satiety, hunger, cravings, and so forth. So this is a very complex system that communicates between the gut and the brain. And in this case, it's directed at the detection of the.
So now we can understand various neurotransmitters which are produced. They generate electrical synapse in the gut which gives signaling to the neurons through what? Vagus nerve. Now vagus nerve passes through various systems and then you can see how vagus nerve keep damaging the organ body and decaying of the body, and we are responsible firstly because of our food. So food allergy is one of the most important thing which we have to consider before considering the treatment of the disease. Food is very important, and if number of chemicals are there which are rejected by the body, for example, you see large number of population they cannot eat brinjal. The moment they eat brinjal, there will be lot of itching in the body, lot of problems are created. So these allergic substances reacted by the body in the gut creates a large number of diseases including neuroinflammation. So somewhere it is also said because of this food what we eat, they create this pathway to the pancreas, and this pathway creates what? Diabetes. This pathway have two cells, beta cells and the alpha cells. So vagus nerve somewhere plays an important role for the insulin production. Now we should maintain our diet in the way that we should not have too many intervals, too much food in the intervals. Why? Because a certain insulin level is induced from the body. We should have a timely food and the induction of insulin in our body, because if you keep on eating several times a day, you have to produce large number of insulin. Again, that is dangerous to our health. Sugar or carbohydrate is to be what? To be converted to a simple carbohydrate molecule which gives energy to the body. So there is a lot of chemical changes which happens from different types of food we eat, and this is done by the bacteria which we have in the gut, and this gut has a brain axis from the vagus nerve. And we have to be very careful while eating our food, because if we don't eat our food in a good way, if the food is not good, we are certainly going to may have certain food diseases in our brain.
Nutrients. But in addition, there are specialized sensory cells, including enterochromaffin cells and tough cells, that are specialized on detecting potentially harmful substances in the food. So this would include various toxins, noxious xenobiotics, and so forth, and as well as some of the infections, including parasite infections. And these two types of cells then, when they detect noxious chemicals, also communicate to the brain and to the rest of the GI tract to elicit appropriate reactions. So if we look closer at how the food is being sensed, there are these different types of sensory epithelial cells, including enteroendocrine cells, tough cells, enterochromaffin cells, as well as just below the epithelial layer there are immune cells and various types of neurons that monitor also what's going on in the gut lumen. Enteroendocrine cells, again, they detect macronutrients, amino acids, sugars, and so forth, produce these gut hormones that communicate with the nervous system and endocrine system. Enterochromaffin cells and
yesterday it was also mentioned.
Noxious
Stop.
substances and produce again.
Yesterday it was also mentioned about the neuroendocrine system, how the signaling from the vagus nerve through the gut enters the neuroendocrine system and also our body endocrine system. It has an influence on both. So that is why if you eat much of the gluten, the sugar will be very high. It was also said by Dr. B.M. Hegre in many of his lectures how gluten toxicity induces high blood sugar inside our body, and we have understood this food allergy creates large number of diseases including the autoimmune diseases. आनंद ये वीडियो उसको भेज देना, क्या नाम है फर्स्टू को, ये
Yes,
2-4
sir.
वीडियो जो है ना उसको भेज देना, ताकि क्या उसको ये समझ में आ जाए वेर ही इज मेकिंग मिस्टेक इन हिज मदर्स केस. His mother is suffering from autoimmune disease,
Yes,
and
sir.
food has become an important part. Maybe he is not taking care of some of the food items which has created a inflammatory disease in the body, right? Especially neuropathic diseases are created because of the food inflammation.
Yes, sir.
Thank you. Carry on.
Various signals, including serotonin, as well as some of the cytokines like IL-25 and TSLP, that then can communicate with the immune system in the gut. And what happens at this stage is what's largely unknown in terms of the mechanisms resulting in activation of the immune system, but detection of noxious substances on the one hand and detection of dietary proteins on the other hand creates these two requisite signals that are necessary to initiate the adaptive immune response. In this case, Th2 response that can result in allergic sensitization. This Th2 driven or so-called type 2 immune response, we think, participates in food quality control. It evaluates food composition and it promotes defensive reactions that meant to minimize exposure to noxious substances. This would be mediated through IgE antibodies recognizing dietary proteins, inducing mast cell degranulation, release of histamine, induction of peristalsis that would promote expulsion of the noxious substances, as well as through activation of C2 cells, production of IL-13 that will promote mucus production, increasing barrier function, and again protecting us from noxious substances. And as you can imagine, exacerbation of these reactions would result in a typical pathology of food allergy. So we were interested in the question of how the brain reacts to these allergic responses in the gut. It's been known since early 90s, mice that are sensitized to a particular type of food, for example, to corn or peanuts, once they sensitize, they avoid consuming that food. We asked whether this behavioral response to food allergy represents a form of associative learning. Associative learning, as you all know, exemplified by its classical conditioning, Pavlovian conditioning, where an unconditional response or pre-programmed stereotypic response to a stimulus, like in this case food, and there would be no response of the same type to a conditional stimulus, like the sound or something else that is not related to food. However, when these two stimuli are paired, then there is an association between them, and that's what results in associative learning or conditioned response to conditional stimulus alone. So that's a typical sort of Pavlovian conditioning. And there is an example of Pavlovian conditioning that occurs in the phenomenon of conditional flavor aversion, or sometimes called conditional taste aversion. What happens here is that if you consume food that contains a noxious substance like a toxin, the toxin itself will result in food poisoning, which would be an unconditioned response to a toxin. So to avoid consuming the same food containing this toxin in the future, what happens is that because we cannot see or smell the toxin typically, but we can detect the flavor of the food that contained the toxin, then what would happen is that the flavor of the food would be associated with the malaise induced by toxin, and that association will result in long-term memory towards the flavor associated with the food in order to avoid potential consumption of toxin in the future. So the flavor now will operate as a conditional stimulus, resulting in conditional aversion of the flavor or the taste. And in this way, the system minimizes the risk of exposure to toxin in the future. And I think what happens in food allergy is very much parallel to that, except that instead of the flavor of the food, what is being detected is food antigens. Because the immune system, in order to react and remember what it reacted to, it needs to see protein antigens. So food antigens are used as a conditional stimulus, and when they are present by themselves, it elicits no response or immune tolerance. When it's present together with a noxious substance that can induce adverse effects, then there is associative learning between the dietary protein antigen and the noxious substance that results in allergic sensitization, and the next time we expose to the same food with the same food antigens, it will result in conditional aversion. Of course, it will result in a normal allergic inflammatory response in the gut, but we hypothesize it also will result in behavioral response of aversion. Indeed, we found evidence for this framework in a few experiments that I'll share with you. The experimental design here is very simple. We use mice that are sensitized to a particular antigen, like of albumin, and then we see whether they exhibit aversive behavior. But first, I'll just show you the control scenario where these mice are not sensitized. So they have a choice of consuming either water, pure water, or water containing the albumin, which they're not sensitized to. So it's not an allergen for them yet. And as you can see, mice actually prefer to drink water with albumin. It probably tastes better for them, so they consume a lot more of albumin-containing water in two different genetic backgrounds of mice, and this is shown in two different ways, either preference for one bottle versus the other, or by the number of times they lick the bottle, it's shown on the right side. So they control albumin-containing solution over pure water. However, once they get sensitized, once they become allergic to albumin, then what happens is that they stop preferring the solution containing albumin, and they start preferring water over albumin containing solution once they become allergic. So that's what it means that they become aversive to food or water sources containing the allergen. And what is particularly interesting is that this aversion is entirely dependent on Ig antibodies. So this behavioral response depends on the presence of antibodies towards the antigen the mice avoid consuming. So this is shown in a couple of ways here. This is comparison of wild-type mice. When they're not sensitized, they prefer albumin, and when they're sensitized, they prefer water. And this preference goes away if they don't have Ig in Ig knockout mice. And similarly, if they don't have Fc epsilon receptor for Ig, they also lose their aversion towards albumin. And this is shown also here using a different type of readout. And interestingly, this type of aversion is very robust and stable. It can last for months. So here we show the comparison again between control mice that prefer albumin versus logic mice that lose this preference and start avoiding albumin. This is 10 weeks after sensitization, and this is over 50 weeks after sensitization. There is still the same phenomenon. There's a long-lasting memory of aversion. And this aversion reaction is actually not dependent on inflammatory response in the gut, as shown here. This is the control gut, normal mucus of the intestine. This is allergic mice that are wild type. You can see a lot of goblet cell hyperplasia, mucus production. And this is Ig knockout mice that also have hyperplasia and mucus production. However, they are no longer avoiding consuming the albumin in the absence of Ig. What we found is that this sensing of allergen in the gut leads to activation of sensory neurons that result in activation of nucleus of solitary tract in the brainstem that then connects to parabranchial nucleus that monitors various sensory information from visceral organs, and then in turn communicates with lateral hypothalamus and amygdala, where the memory of this experience is stored. So this gustatory, olfactory, and chemosensory information from the gut goes to the nucleus of solitary tract that collects all this information, transmits it to parabranchial nucleus, where this conditional taste aversion and adverse reactions to food recorded, then transmitted to lateral hypothalamus and to amygdala to induce aversion and anxiety, which is also present in human patients that have food allergies. So this is an example of gut-brain connection where the sensory neurons that monitor the gut and connect to the brainstem actually get activated not directly by stimuli from the gut lumen, but are activated through Ig antibodies that recognize allergens. And Ig antibodies then get deposited on mast cells. Once they recognize the allergen, leads to mast cell degranulation, release of histamine, neuropeptides that then activate neurons, leading to the activation of those brain circuits that I just showed you. So this is an interesting variation on the theme of how neurons detect various stimuli. They can either detect them directly, as is the case with olfactory neurons, or they can detect them via specialized epithelial cells, as shown here for visual system and auditory system. And here we now have an example where it's stimuli detected indirectly through antibodies that hit on mast cells and induce the activation and release of neurotransmitters that activate neurons. In conclusion, conditional allergen aversion is a version of conditional taste aversion, except that instead of flavor or taste, which is being detected at food allergy, and that results in a very similar type of aversive behaviors. Allergic sensitization results in allergen avoidance, which is the behavioral defense against consuming noxious substances, and it's Ig mediated and involves this brain circuits that generally involved in defensive behaviors. Finally, this work was done by a very talented postdoctoral fellow, Esther Florsheim. She was helped by a very talented research associate, Jamie Cullen, and Fernando Carvalho assisted with some of the experiments. And two collaborators, Marcelo Dietrich and Daniel Nisida, helped with various aspects of the project. And this work was supported by Food Allergy Science Initiative and HHMI. Thank you very much for your attention. Hello, I'm Ruslan Majitov. Today I will talk about
Sastri ji? It looks like Sastri ji logged out or got logged out by the network on his area. Let's see if he'll come back on soon. He was trying to log in, and I don't know why he's not yet back in.
Maybe just wait. We can waiting for Sastri come in to the back to the class.
Yes, a few minutes. Let's wait. He was trying to log in. I don't know. Maybe he's still having network issues too.
Thank you, sir. Dr. Anand, please call to sir.
Okay, he's trying to join in again.
Sorry, it was disconnected because of the power cut off. So now you can ask me your questions. The lecturer here was focusing on the food poisoning and food allergy. Now you can understand how important is the food allergy, and the use of antibiotics creates lot of damage in the body, because good bacteria are damaged. Bad bacteria are also damaged and good bacteria are also damaged. With the damage of bacteria in the body, you can understand lot of inflammation takes place because of the what? Signaling transferred from the vagus nerve to the brain. Now you can understand how depression is created. Now you were saying me the root cause, root cause, root cause. Now we can understand very well what is the root cause of the what? Diseases. Root cause means what? From how it is created. So symptoms are nothing but the pathway to the disease. From symptoms you understand what is the disease. The disease will say how it was created. So when you analyze the disease, you will understand which kind of bacteria was involved in creation of this disease. The inclusion of bacteria will allow you to understand which kind of food has created this disease, and the involvement of gut. Yesterday, a very good environment took place. Walid, can you go to my Facebook? I want to show you one very good video which we have taken yesterday.
Yeah, let me try and get this.
You will see a patient who came in a Glasgow coma scale coma patient who was non-reactive to any of the external stimuli. Means if you give pain or anything, she was non-reactive to it. And after the treatment, you can see the patient is walking. Generally, patient who has hyponatremia are dehydrated patients, and their sodium level is very low sodium level. So even if you correct it to a good sodium level, by then there is damage in the bones, and these people go to a coma, and they remain in a vegetative stage. But you can see in this case the patient has revived soon, and it has responded to all the external stimuli. The classical coma scale is 15 by 15, and it is very difficult to revive these patients. These are rarest of the rare case which has been revived by us in a small period of 20 days, whereas the patient has already spent more than 1 month in ICU. So, Glasgow coma scale 3, 4, and 5. Finally, Dr. Avinash is the physician of the patient who will put more light on the patient.
Yeah, hi, this is Dr. Nash, myself physician. She was admitted with severe hyponatremia at our hospital, Lotus View Hospital, Tirunelveli, Hyderabad. So with severe hyponatremia, so she came with altered sensorium with Glasgow coma scale 3 by 15. She was completely unresponsive. She was not responding to any verbal commands, pain or nothing. So the reason for this unconsciousness was severe hyponatremia. So we took a MRI scan of her. It was showing central pontine myelinosis. So we have corrected the sodium, and sodium has come to normal in 2 to 3 days. Despite correction of sodium, patient was still comatose, and GCS was around 4 by 15. So we have explained the relatives that the recovery is less likely. So through my contact, I have referred to M.K. Shastri sir. So sir has started treatment since 1 month back. So she has responded well. She started responding to verbal commands. First she responded to pain, then she slowly started responding to verbal commands. And today miraculously she started walking. So
How much chances are there of walking of such speed?
less than 1%. Even neurologist has explained that she'll be in vegetative state for long months, long years maybe.
And within 20 days, if she starts walking, what will you say about the excellence of the treatment?
It's completely excellent, unexpected, miraculous recovery.
Please make the patient walk. 13 Martin March. एक महीना भी नहीं 20 days he started walking. So can such patients start walking?
No sir. We've seen many patients of central pontine myelinosis with long-standing bedridden status and low GCS and all. This is kind of very excellent recovery.
With dry needles, is it possible?
It's completely miraculous.
So now she can walk. You can see. Hey, walk around. Now, what is her glaucoma scale? She came by 4 by 15, and now you can say in 22 days she has become what? And our Bedford also started healing, which was so we thought of plastic surgery.
కమలా గారు చూడండి.
Now do you think rotational therapy is required? So put some more light on this patient.
So even she has a very deep bed sore. So we thought of planning a plastic surgery, rotational flap. So even I her bed sore is started recovering. No tissues growing in the bed sore. That is kind of good recovery. So even she started walking. Hope she will recover more. Hope she will develop some speech in nearby future.
No, but she has started responding to speech. She has started speaking words. Hands have started moving. So over glaucoma, carried with 4 by 15 minutes.
Yes.
And now GCS 15 by 15. Then hyponatremia, do you think such cases can be covered when phones are affected?
Less recovery rates.
Less recovery rates.
Even I have many patients. They are bedridden since 1 year, 2 years.
Vegetative stage.
Vegetative stage.
But in 20 days with dry needle stimulation, what do you say?
Completely unexpected recovery, I can say.
Completely
miraculous recovery.
You can see the patient has walked up to that level. ले जाओ उसको ऊपर ले जाओ. So in 22 days, how do you find this recovery? How do you find? You have been the physician to the patient.
Initially, when I see the patient initially, before 20 days, she was just bedridden. She was not even responding to commands. So in the first week, she slowly started babbling and responding to pain started. And
now she is walking, what will you say?
It is unexpected for actually.
Unexpected recovery.
Even I didn't think that she recovered so soon.
And treatment, you think is very good. Very good.
Actually, I said relatives that she will recover in 3 to 4 months. Duration is very less. That I can see.
Duration which we took less than 20 days.
That is very, very fast to recover.
This such type of patient.
For this case.
At least for such type of case. So such patients can be recommended for this type of treatment.
Obviously, I recommend.
So here you can see a case of hyponatremia, glaucoma scale 4 to 15. Within 20 days she came out from coma scale 15 by 15. ये केस था hyponatremia का जो कोमोटोस था यानी कोमा स्केल में था. पेशेंट वाज नॉन रेस्पॉन्सिव टू एनीथिंग, नॉट इवन टू पेन. सोडियम लेवल करेक्शन होने के बाद भी सोडियम लेवल करेक्शन लेने के बाद भी पेशेंट वाज नॉट रेस्पॉन्सिव. He was treating the patient both in lotus and ear, and every day he was on observation. Treatment was done by our team, and we could see jointly we treated the patient, and we found that patient has recovered in just 20 days, very significant level. Today our scale, आज का scale glaucoma scale is 15 by 50. Thank you so much.
She has been diagnosed as central fountain myelosis in the
Central
month of December 2021.
Close it. Close it. So, do you understand the case where central pontine myelosis takes place? And if this takes place because of hyponatremia, the patient becomes almost difficult to be recovered. You see them years after years on vegetative stage, and we recovered this patient from glaucoma scale 4 to 15. Whereas the previous neurologist who have examined the patient has said that this patient cannot recover. Only by giving a good feeding and tapering antibiotics, we have got good results in the patient. कौन? क्या लाया? बट हम तो इतना जलेबी मीठा खाता नहीं है बस. Now you may ask me your questions. So you asked me how brain stimulation recovers a patient. You can see how conservative medicines when gives up, there is still a ray of hope of recovery of the patient, and it is found in. Thank you. मेडली बस इतना डायबिटीज की हम More questions, please. You can ask me your questions. Can you hear me? आप सवाल पूछ सकते हैं. Ask questions, please.
Go. Alis ako mga banda alas 12. Pauwi na alas 10 na.
Ask me your questions. So now we can understand if the pH balance of the water is not there, frequency of urine sepsis, urine infection is there, lot of other diseases occurs in the patient. Getting too much artificial sugar also is very dangerous, and eating too much spicy food should be avoided because it is not good for our digestion. So any more questions?
सर क्या वेगस नर्व स्टिमुलेशन से हम अपने गट को सेव कर सकते हैं?
नहीं, देखो वेगस नर्व स्टिमुलेट करना इतना आसान नहीं है, उसके कंपार्टमेंट्स है. That is where the acupuncture plays a role. Otherwise आप एक वेगस में नर्व स्टिमुलेटर इम्प्लांट कर सकते हैं in epileptic patient and all, but the prognosis is very poor. I have seen them bringing dead bodies to my clinic. Means the patient is alive but is almost in the coma scale. So this dry needle based stimulation is the key source of information, especially tongue acupuncture, by which you can needle the distal area and react in the SI area and other areas of the brain, SI area and other areas of the body, which directly and indirectly stimulates the vagus nerve. Signaling the vagus nerve, you can understand how stimulus is produced in the brain. That is why it is said reaction nerve and reaction area. It gives signals to the brain, brain reacts to the area, area reacts to the brain, brain reacts to the point. It is like a triangular system like this. Toi, toi. First going like this, you needle here, it goes like this. Then the reaction nerve goes, reaction is forwarded here. From here it goes back to the organ. Organ again it stimulates, again it goes to the brain, and again it comes to here. So this signaling continues until 45 minutes. And then the outcome of tongue acupuncture can be seen. And this is how brain stimulation helps you to recover a disease, especially like paralysis and all. It stimulates what? The certain nerves. And stimulation of nerve stimulation is very easy through the acupuncture nerve process of system. Many such papers have been published and many such things have been seen in past. Questions, please. Questions?
So that means through needling, or we can say tongue acupuncture. We process the command through the afferent, and then the brain interpret the signal, and then a motor command will take place.
You are right. Similarly, we do it with the brain stimulation. Sensation creates a motor command. We use the pyramidal way. But in this case, you could see central pontine myelinosis was the most difficult because the pons of the patient was affected. The pyramidal pathway, which is the motor and sensory pathway itself, is affected. When the neurons of the pons are affected, then you can understand the patient is difficult to recover. And the neurologist said, even the medicine doctor was saying me, it will take months and months and years to recover. So Sastriji, you should have a party. I said, no, the patient will revive very soon. You just give the chance to revive for the revival of the patient. And you can see, patient revived. That is what I said. It is much to be learned and much to be understood before recovering a patient. और बोरा जी अपना चेहरा तो दिखाइए जाने से पहले.
एक मिनट सर.
अरे वाह कितने प्यारे लग रहे हैं. ठीक है बेटा, अच्छा लगता है. एक बार देख लेते हैं तुम लोग को तो सुकून मिलता है. वेरी गुड. थैंक यू सो मच फॉर द काइंड कंसिडरेशन.
Thank you, sir. Thank you, sir.
So now you can understand how you were asking me how NIBS helps. NIBS helps both in recovery of the gut and also recovery of the brain. It helps you in neuroplasticity of the brain also. It helps you in the recovery of what? Various things can be done by signaling. If vagus nerve signaling can be, if gut signals the brain, brain also signals the gut. More questions? More questions? More questions, please. Any more questions?
May I ask a quick question?
Yeah. Please ask your question, please.
What do we use in place of antibiotics to treat patients?
See,
that
here
so
the infection level is very high when the patient comes to us because the immunity level is low. But we use low power of antibiotics as well as probiotics. Prebiotic, probiotic, we use it, and then we put the patient on multi-IV therapies. So somehow or the other we try to revive the immune system of the body. First we try to reduce the deficiencies of the body. Second we try to revive the body. And then we try to revive the brain. In this case we did not use very hard antibiotics. You could see this patient was a rare example where it was recovered within few days of the treatment. You start improving from Glasgow Coma scale from 4 to 8, 8 to 9, 9 to 10, 10 to 11, 11 to 12. Now you can see the patient is on M6 level. Motor function is 6. So you could see a good recovery in this patient. So we must understand that only the needling is not sufficient. Knowledge of medical science is most important. And medical science ka knowledge is very important for the patient.
Yes, sir. Thank you.
More questions, please. More questions? So questions are not there?
Good evening, sir.
Yeah.
Sir, as said, you told that gut inflammation causes gray hair. So gut inflammation causes gray hair. So can it be cured by dry needles?
See, we have to see what kind of infections we have and what has been damaged.
Okay.
For example, conservatively, very little could be done in this case, where central pontine myelonosis was there because of hypernatremia. Hyponatremia, right?
Yes, sir.
So, we thought that she will be continue to be in the stage of vegetative, where her sodium was corrected in 2 or 3 days. We thought that the patient will revive, but then they did not revive because they had central pontine myelonosis. This is where the brain starts shrinking and the pathways are damaged. But still then she revived because of the pathways could be recovered. So what was used? Brain stimulation, nerve stimulation. That's all.
Yes.
Antibiotic has its own role in infections. But in such cases, we have no drugs to revive the patient. We can only do corrections.
Yes.
So sometimes we must understand that body can manufacture its own drugs. Allow the body to manufacture its own medicine. Always putting the patient on medicine is not correct. Allow the patient to be on his own medicine. Am I correct?
Yes, sir. Okay, sir. Thank you, sir.
More questions? This is why I said having a good knowledge of acupuncture and integrative medicine will only help you to revive the patient faster and quicker. More questions? Can we have any more questions? Okay. Since there is no questions, I'll take your leave. Thank you so much.
Thank you, sir.
Thank you, sir.
Thank you,
Good night,
sir.
Good
Thank you,
Good
sir.
night, everybody.
Thank you so much,
Sorry
sir.
to
Thank you.
this.
Appreciate you.
Thank you all.
Good
Thank you,
night,
sir.
everyone.
Good night,
Thank
everyone.
you.